Okay, so let's talk about the main blood cell groups. We have erythrocytes, leukocytes, and thrombocytes. Erythrocytes are what you're seeing in this image are these sort of pinkish with a lighter area in the middle, or red blood cells. So our technical term for red blood cells is erythrocytes. Leukocytes are these larger cells that are kind of bluish purple, somewhat grainy or somewhat odd lobe. These are our white blood cells, and these three shown here are different types. And then thrombocytes are these tiny, tiny little dots that you're seeing here. Those are our platelets. So again, these are our scientific anatomy terms for red blood cells, white blood cells, and platelets. So let's talk a little bit more about erythrocytes. What you're seeing here is a general main function of red blood cells, and we'll talk a little bit more about this too when we get to the respiratory system. But red blood cells, I think we recognize this, they carry oxygen. They are going to deliver that as well, and nutrients. They're going to deliver that to the body cells. And let me just back up what I just said. The red blood cells are carrying oxygen. The plasma will be carrying the nutrients. So just so I'm saying this clearly to you. As blood travels in the capillary, again, the red blood cells never actually leave the capillary. It's just the fluids that's leaving. And what you're seeing here at the capillary level, you're seeing it's turning from that red color to a purple to a blue. That denotes that it is lacking now oxygen. It has delivered all of the oxygen that it was carrying to the cells. At the vein side of a capillary, it is now picking up carbon dioxide as well as waste, and then it's taking it back to the heart to pick up oxygen and to the kidney, and then have it go back into systemic circulation to send it to the kidneys to get rid of the waste. All right, a little bit more about red blood cells specifically. They are a biconcave disc, so all that means is that it's one curve in the disc as well as a second curve on the other side. They're tiny. They are 8 microns in diameter, pretty uniformly 8 microns in diameter. They do not have a nucleus or organelles. So I want you guys to think about these 3 questions for a moment. So I'm going to just pause. Maybe you can hit pause on the recording too. Just give it a thought. So what does it mean that a red blood cell that has no nucleus or organelles? What's the benefit of that? And then what's the downside? Okay, so hopefully you paused the recording and you gave it some thought before listening for the answer. So when you don't have a nucleus, so think about it, the nucleus is the part of the cell that holds DNA. And the whole point of having DNA is that you can transcribe it and then translate it into proteins. And you would need to do that if you're making more of the cell or repairing parts of the cell. Now, so that means for a red blood cell, it doesn't have all the organelles or a nucleus, so it doesn't have your rough endoplasmic reticulum, ribosomes, Golgi apparatus, all of those things that help to make proteins either. So what that means is it has no way to repair itself. So as it ages, as things break down, it can't fix that. So red blood cells are going to have a finite lifespan. So that's one of the downsides. One of the benefits of this, though, is that whenever you are building parts of a cell, building proteins, guess what? You're using up oxygen and nutrients, right? Well, if the whole job of a red blood cell is to carry oxygen, you want to make sure that it is not consuming the oxygen that it is carrying, right? So more of it can get to the cells that need it. So the benefit of not having organelles or nucleus is one, that it doesn't have to use up the oxygen that it's carrying, so more can be delivered to the cells that need it, as well as by getting rid of all those organelles and nucleus, it ends up having this biconcave disc shape to it, which means that it's kind of floppy in the middle, so it can actually squeeze through some tight spaces as well. So it allows for flexibility and more efficient oxygen exchange. The oxygen-carrying protein that is called hemoglobin. It gives blood its red color. It's kind of a cool picture. It looks like some bowl of red Cheerios to me. And it is a good portion of what is an actual red blood cell. So it's about 1/3 of the weight of the cell. So this is what hemoglobin actually looks like. So hemoglobin is a polypeptide molecule. So you have these 4 chains. Each one of them has a protein part, which is called globin, and then there's a non-protein part called heme. And that's this sort of Saturn-y looking thing here in the middle. That heme has an iron molecule in the middle of it, and that's what allows oxygen to attach. Okay, so each hemoglobin molecule, each one of these, can attach 4 oxygen molecules. Okay, and then one of these red blood cells have about 280 million hemoglobin molecules. I don't even know what that number looks like. That is such a large number. But think about it. So if each red blood cell has 280 million hemoglobin molecules, and each hemoglobin molecule can carry 4 oxygens, 4 times 280 is, I don't know, a billion something, a lot. A billion something oxygen molecules can be carried by just one red blood cell. That's pretty impressive. Okay, so how do we actually form new red blood cells? Okay, so the process is called erythropoiesis, and the main stimulus for the formation of new red blood cells is hypoxia. Hypoxia is basically lack of oxygen, or very low oxygen levels. That's hypoxia. The protein, or I shouldn't say, well, it is a protein. The protein and hormone that stimulates red blood cell production is erythropoietin. Okay, so that's what this flow chart is showing you. So some stimulus is disrupting homeostasis by decreasing oxygen delivery to the kidneys and as well as other tissues. Your kidneys, however, because their main job is to filter blood, they actually have some special cells in them that are monitoring oxygen levels. We'll talk a little bit more about kidneys too in another section. But kidneys are going to detect these low oxygen levels. They're going to send signals, as well as they'll send some signals to the brain, letting you know that you have low oxygen levels, as well as increasing erythropoietin that's being secreted into the bloodstream. Again, that's that protein right here that's going to then stimulate these precursor cells in the red bone marrow to mature more quickly into something called a reticulocyte. A reticulocyte is simply an immature red blood cell. That's going to send more of these immature red blood cells into circulation. Those quickly mature, which then forms more actual mature red blood cells. More red blood cells means you're going to have an increased oxygen carrying capacity to your tissues. And you go back to homeostasis. So a typical environment where you would see this in, this would be what happens to athletes who train in high altitudes. At high altitudes, there's less oxygen. So again, the kidneys would pick up on that. They would send signals also to the brain saying like, hey, we're also getting low oxygen, so increase our breathing rate, but we'll talk more about that part when we get to the respiratory system. But they will also increase the production of this erythropoietin, which is a hormone that then sends signals to precursor cells in the red bone marrow to mature more quickly, produce more reticulocytes, which are again immature red blood cells, send those into circulation, those quickly mature into mature red blood cells, and then we can increase our oxygen carrying capacity. Okay, so again, these athletes that train at high altitudes, what they're doing is they're increasing their red blood cell levels, or I guess ratio in their body. So then when they actually compete in a competition, they have more red blood cells and more oxygen carrying capacity, and therefore fatigue. It takes longer for them to fatigue. Okay. So again, the formation of your red blood cells, the erythropoiesis. So after a nucleus is ejected, we call that a reticulocyte. Those reticulocytes still have some of their organelles, so they're not a fully mature red blood cell yet. You can see a reticulocyte is orange in color instead of that nice red, and there's still some traces of rough endoplasmic reticulum. Remember, that's the endoplasmic reticulum that has ribosomes on it. Ribosomes are what produce proteins. But once they are a reticulocyte, they can enter the bloodstream, and very quickly, within 48 hours, they eject the remaining organelles, and they become mature red blood cells. But as we mentioned, because they've now ejected that rough endoplasmic reticulum, they're no longer able to fix any damage that may occur, so they age out. So they only live about 120 days. They wear out again, that biconcave shape allows them to bend and flex, but after a while that breaks down. So any of these cells that are worn out are removed by macrophages in the liver or the spleen. When a macrophage, so this is a cell that will gobble up other cells, as you're kind of seeing in this cartoon, when it stays in one spot and it's not moving around the body, like these are just living just in the spleen or just in the liver, they're called fixed macrophages because they're in a fixed location. And then the products, the macrophage will break down products and reuse those in the body to produce more red blood cells. So what does this process look like? Now I'm going to go to the next slide. It's going to be pretty busy, but I will lead you through it. Okay, it's actually simpler when you break it out. When you put it all together, it's a lot of steps. Okay. There's also numbers here, and that might be helpful to kind of look at it in this sort of numbered fashion as far as like a pathway. So we'll go through it. We'll use the numbers to help guide us. Okay, so we'll start here. We just produced a red blood cell. It goes into circulation for about 120 days, right? Like we said a second ago. And then it wears out. Okay, well, here we are. This is a macrophage in the spleen or liver, and it's possible for them to be in the red bone marrow as well. I didn't include red bone marrow in the prior slide. But here's a macrophage in one of those locations. It will gobble up that red blood cell. And then let's follow this pathway. Part of that, remember we said a third of the red blood cell is hemoglobin. So the globin part, this part, is just protein, said a couple slides ago. So the building blocks of proteins are amino acids. So the globin is simply broken down into amino acids and then reused to make new, maybe the same, maybe different proteins. Okay, so if you think of red blood cells as primarily hemoglobin, the globin part is very simple. Globin, protein, amino acids reused for protein synthesis. Now the heme part's a little bit more complex. Remember that heme was that Saturn-looking thing with the iron molecule in the middle. So let's see what happens with the actual iron. So the iron will be transported and predominantly stored in the liver for future red blood cell formation. Now iron is because it holds on to oxygen so tightly, it can be very damaging in the bloodstream at high levels, or if left unbound to something else, it can actually start breaking up bonds. So whenever you're transporting iron, it actually has to be bound to another protein to basically keep it inactive, to keep it kind of from hurting, damaging other parts of the body. So the protein that it's bound to, it's called transferrin. So iron is just ferrous, right? That's the Fe. So think of it as a transfer into the liver, if that's even easier, but it's transporting the ferrous ion. In the liver, it is bound as ferritin, okay, as a molecule called ferritin. Again, you don't want it to damage the liver. And then once it's needed in the red bone marrow to produce more red blood cells, simply we have to attach it to that transferrin, that shuttle, and then take it to the red bone marrow. So again, these iron molecules that you have in the heme, remember that was that Saturn portion of, that center portion of the Saturn of heme. The iron is transported via transferrin to the liver, where it's stored as ferritin, where again it has to be then transported to the red bone marrow for new red blood cell production. So again, simply attached to transferrin. Okay. But what about that outer ring part of the Saturn molecule? Well, or that Saturn molecule that is heme. Well, that as the iron is removed from it, it becomes something called biliverdin, because it's kind of got a greenish color. It will actually be some of the stuff that gives bile its color. So this biliverdin is fairly quickly converted into something called bilirubin. Bilirubin is also transported to the liver, or I guess stays in the liver if the macrophage happens to be in the liver. That bilirubin is secreted, and you can see this here in bile. So The liver produces bile. The gallbladder will store the bile. Bile is secreted into the small intestine. As it moves from the small intestine into this large intestine, bacteria in the large intestine convert bilirubin into urobilinogen. Some of that urobilinogen will be transported in the bloodstream to the kidney, where it is secreted as urobilin. It's converted one more time. And if you think about this, urobilinogen, that ogen, whenever you see that, that just means it's sort of the inactive form. It's like a precursor form. So urobilinogen in the large intestine, urobilin, just get rid of that ogen part in the kidneys. And then that's also what gives urine its color. Okay. The urobilinogen that stays in the large intestine that isn't transported to the kidneys is converted into something called stercobilin, and that also is what gives feces their signature color, if you will. Okay, so just break this down to steps for an exam for you to remember. So this is one of our long processes or pathways in life sciences and anatomy. Again, we had hemoglobin. Globin is just a protein. It's broken down to amino acids that are then going to be recycled to make other proteins. That heme, that Saturn part, the iron part, is stored in the liver. And then when it's needed again, it's sent to the red bone marrow. When it's stored as ferritin. When it's being transported, it has to be transported using transferrin. The remainder of that heme molecule is, once you remove the iron, is called biliverdin. That's quickly converted to bilirubin. It is secreted in the bile into the small intestine. In the large intestine, bacteria will break that down or convert it into urobilinogen. Some of that goes into the blood. Some of that urobilinogen goes into the bloodstream and is taken to the kidneys, where we just chop off that ogen part and becomes urobilin and secreted in urine, again giving its color. Whatever remains in the large intestine is further converted into stercobilin, and then it is excreted in the feces, again giving them their color. Okay, I will stop there. Hopefully that made sense. If you have any questions, please let me know.